clock is regulating metabolism, but it's metabolism now regulating the clock. And I think we're going to find more and more of these examples in the lab, and I'm sure other people are doing this type of studies too. Now, I'd like to finish by thanking the people in the lab. This is not the campus, but it's only 5 minutes from the campus. It is called Crystal Cove. It's a very nice state park. And this is where we work. And I'd like to thank Yazoo, Kristen, Saurabh, and Ben for all the work they've done. Thank you so much.
Very nice talk. So for the BMAL1 installation, did you check whether other ester transferase can also target the same lysine site?
That's a very good question. What I can tell you is that if we do that experiment in vitro, yes, but when we take cells that we have generated where the hat activity clock is dead, that acetylation is gone. So I believe that while technically it could happen, the critical hat doing that acetylation is clock.
Hi. I was just wondering about maybe, does clock end up in the cytoplasm at all? So are there metabolic targets, so proteins that are directly acetylated by clock to sort of, as you said, rheostat the crosstalk between the circadian and the metabolism?
That's a very good question. Clock moves into the nucleus very fast. So I believe that most clock targets will be nuclear. However, I would say that we have the feeling that maybe the short time is in the cytoplasm is going to do something. The reason I'm saying that is because we feel there is at least one cytoplasmic target for which we're almost sure about. So I think it's a really interesting question. Yeah.
So is this circadian rhythm alive in the regular cell lines we're using?
Yes.
So do we have to normalize for this circadian rhythm while making experiments?
Let me tell you this. So it's for the audience. This is true for sure in the animal. Whoever works with mice, mice are nocturnal animals. Every time you do experiments during daytime, you bother them a lot. And so your level of stress, not your level of stress, but this level of stress of the mice is going to increase. Anything you do that have to do with inflammation or responses, behavioral responses, you be very careful. So that's one thing I want to tell you about mice. In mice, for sure, the whole system is circadian. In the cells in culture, it is circadian. The problem is that unless you use those suprachiasmatic nuclei neurons that are all nicely synchronous, the fibroblasts in culture or any type of cell in culture lose the synchronicity. If you take each single cell, that will be oscillating for 50, 60 cycles, which is 60 days in culture. It's forever, basically. But what they don't have is the synchronicity to be all together. So what you do when we do an experiment of serum shock or a DEX treatment in cells in culture, we see a beautiful oscillation by 2-3 cycles. And then after those 2-3 cycles, the cells lose overall as a plate oscillation. But each one of them will still oscillate.
Yes, I think this could be a major cause of the variations in something like measuring histone acetylation, and in one day, the second day.
I personally believe so, yes.
Okay, so do we have to, what should we do about that?
I think you should redo all your experiments along a circadian cycle. Okay. Good luck.
Hi, so similar to the SIR2 of yeast, which is part of a couple of complexes, like the SIR complex and the REN complex, SIR-T1 was shown to be in a complex as well, in, I believe it was in mice. So I wonder if you think the functions of SIR-T1 in circadian rhythm involve any of other members of the complex, such as SOF, I think, one of the SOF39H1? Or
all right, so the question, I believe, is whether in the clock SIR-T1 complex there's anything else. of interest that has to do with with chromatin remodeling? The answer is yes. So we've done a mass spec with clock, and I can tell you that there is 2 more molecules of interest, that I'm sure to this audience, that are directly involved in the circadian function. That are all, both are regulating the chromatin, histone modifications.
So is SOV one of them?
Sure, I'll do something about this. Maybe. Is that okay? Thin chance.
I have a question here. I'm not giving away any secrets when I say that the PSC2 complex was shown by Glenn Grimes at the 31.
Yeah.
I just wondered, this may seem not relevant, but I just wondered if you know whether 13-1 activity or NAD plus level are correlated with temperature.
That's a one. Yes. So Dr. Pirata is asking whether the SIRT1 activity and NAD plus levels might be somehow modulated by temperature. Well, that's a great question. And the reason why it is a great question is because I believe so. And also because there is one thing I didn't show in my slides because of lack of time. There's one thing, one way you can re-entrain circadian clocks besides light, and it's called temperature compensation. That's what we all do, no matter what. Without even knowing it, we're doing it by putting ourselves under cover, and because our thermoregulation, our homeostasis is requiring it. So I believe so, and I believe that, temperature is gonna be critical. I'd like to just mention one last thing, is that one of the transcription factor of the Hitchcock machinery, HSF1, is, I believe, is going to be shown soon to be a critical circadian regulator too.
Is this also case for mitochondrial genes?
What was the question? You're talking about the oscillation? Okay. You know what? Is not well known. I mean, the thing is not clear. Okay, that's the real answer. Some people say yes, some people say no. The fact is that maybe that's where you're coming from. You know, SIRT3 is in the mitochondrion and is regulating the energy balance within the mitochondrion. I can only say that if your question is whether the other way around, if mitochondrial function is important for circadian responses, we're doing experiments with Doug Wallace on this. He has a number of knockouts for specific mitochondrial genes, and the answer is yes.
Okay.